Name of his talk today for the Dwyer Lecture, the Philodendron family, Aroid, diverse, interesting, and still growing. Dr. Croat, if you'd please take over.
The Philodendron family, of course, may be better known to some of you as the jack-in-the-pulpit family. It's a local forest plant in Missouri. I've spent the last 50 years working on aroid for the most part. I came to the Missouri Botanical Gardens after writing a letter to Walter Lewis, who was then director of the herbarium, just simply asking, do you have a job working with Flora of Panama? Because I saw these wonderful treatments in the library at Lawrence. And I'd never been to St. Louis, nor had I really heard of the Missouri Botanical Garden to any great extent, but that looked like an intriguing possibility. Walter Lewis wrote back about a week later saying that he did not have a job working with the Flora of Panama, but that there was a job working to write a flora of Barrow Collar Rattle Island. And I was very interested in this prospect, so I came over. As far as I know, he never interviewed anybody else. I convinced him that I could do the job. On Barrow Collar Rattle Island, I set to work going down there every 2 months on, 2 months off. So I was down there for 2 months, came back for 2 months. I did this for 2 years so that I covered one entire year. The objective was to see every month because I was very interested in making sure that there wasn't a month when there was some plant lurking in flower that I had not seen. The island has many coves and inlets, and I worked around the island with a boat to collect in these inlets and coves and bays. But I also spent a lot of time collecting plants. Hundreds of trees I had to climb with climbing irons. And to get up into the top of these trees and see out over the top of the canopy was quite an exhilarating experience. Oftentimes you'd get up in the tree and you'd realize that the tree you were in wasn't the tree that had the flowers falling to the ground. I usually tried to concentrate my efforts on the pattern of flowers, and I'd climb the tree that's sort of in the middle, and I'd go up there. But oftentimes it was in the next tree. And when I could, I just switched over into the other tree going through the branches. Very often I would go up one tree and come down another one because I would find that the other tree was a little bit more suitable for going down. I always carried 50 feet of rope with me so that if I got to a situation where I couldn't get down, I could lower myself with the rope. I also, at that time, began collecting plants in other parts of Panama, and I was especially concentrating on aroid because I found that on Barrow Collar Rattle Island, these were very complex and confusing. Because of this, I had to learn all about the aroid myself. There were no experts, and then as I went around Panama collecting, I realized that everywhere I was going, I was actually finding new species. I did not realize it at the time they were new, but nearly all of this stuff turned out to be undescribed species. I had opportunities to collect, like when this trip in these dugout canoes to rescue animals on Lake Bayano. I went into a gold mine with Tyler Kittredge to collect in his area. I went with Flora of Panama expeditions that were relaunched by Walter Lewis, like one trip here with Dr. Dwyer on the Santa Rita Ridge Road. This picture in the middle of the assemblage here is kind of interesting. That's a picture of John Dwyer at a little hut in Belize. This guy had these little shacks that he had let us collect in, and one day when I went up to the bathroom, I came back down. When I was up there was an explosion. And what had happened was our little pot white gas stove blew up, throwing gasoline all over the place, and the whole thing was in fire. Being a farm boy, when I came back down, Dwyer and everybody had run outside. Being a farm boy and having a lot of experience with gas fires, you run into the fire, you don't run away from it, because otherwise everything is lost in a matter of seconds. So I kicked this dryer out into the yard and put it out. I've done that many a time. All the while I was collecting plants in all these different places, I ended up collecting a lot of aroids and bringing them back to the Missouri Botanical Gardens, oftentimes 1,000 cuttings at a time. Of course, they didn't all live, but a great many of them did live. And we eventually, the weak ones eventually die off, and you end up with the plants you've got. The living collections have been used for a variety of different purposes. I'm going to back up one here. They've been used for a variety of different purposes for other people's studies, cytological studies, anatomical studies, DNA studies, even breeding studies. I wanted to mention specifically what is an aroid, because I've talked about aroids. An aroid is a common name for the family Araceae, just like orchid is the common name for Orchidaceae. So it's nothing but a common name for this group of plants in the Araceae. And they have, the only unifying character really is that they have a spathe, this leaf-like structure that initially encloses the spadix, and the spadix is a spike-like structure like a corn cob or a cattail, if you understand what that is, and the flowers are all aggregated on it. So they're very tiny flowers, not like a rose or even a dandelion. They're very small flowers, and they're aggregated on this spike, which we call a spadix. And you'll see that some of these common names are things that you are familiar with, things that you can find in Kmart. The Araceae has some very important characteristics. First of all, it's one of the families with the highest species diversity of any that I know. It also has high habit diversity, that is to say, what it looks like, how it grows. Epiphytes, terrestrial plants, floating plants, and so forth. I'll talk about this more in a moment. It has high rates of endemism. In other words, these plants very often occur only in one place or only in a few places. All throughout the tropics, every hill that is forested has some aroids on it. And it's one of the reasons why I am urging conservation, because you just take off one hill or cut off one river valley and you're taking aroid species that are endemic. Secondly, or thirdly, I guess it's fourthly, undescribed species. The family has massive numbers of undescribed species. I'll give you some statistics on this later and point out how important this is to my studies. Why are there so many undescribed species, and why are there so many endemics? I think it's because Anthurium has co-evolved with euglossine bees and some other insects. These are also high in species diversity. So the highly diverse insects and the highly diverse plants go hand in hand, and they keep pushing evolution. The other thing is that aroids tend to be dispersed by birds in the forest. And these birds are not migratory birds. They're terrestrial. I mean, they're birds that live within the ecosystem, and they're often territorial. They don't move very far. So if you have a mutant species, it doesn't get carried very far away, and yet it can be very plentiful in one place and never exist in another. The same thing is true of those beetles. The beetles are very high in diversity. Now, I want to give some examples of high species diversity. The picture in the upper left where it says Cerro Piri, or Cana gold mine, one of the first places I went to in Panama was this gold mine, old gold mine. Texaco had an opening there. And then the price of gold dropped and they closed camp. But they still had this airport so you could get in there. In a single day, I collected 267 collections. 40 of them were aroids, and 10 of them were undescribed. And this could go on almost anywhere you go. The same thing could happen in many, many other places. Other examples just like it are, for example, in Bajo Calima, or in the next picture down at Lita San Lorenzo, Alto Tambo. It's absolutely reeking with plants. And I spent about 8 trips in that region around Alto Tambo, and then one time I decided I'd go to El Chical, which is in another river system only 50 kilometers away. I was shocked to find out that everything there seemed to be unrelated to the things in Lita San Lorenzo, and yet it was very, very close. And this same experience has happened to me again and again as I look at close by places, finding that the flora is very dissimilar, even though they're the same life zone, same elevation. Very high species diversity, and it's quite amazing. Then the same thing was true in Bajo Calima, where I did study with Dorothy Bay. And even most recently, a couple of years ago, I was in the Rio Medellin, near the city of Medellin. I collected about 45 species, and of those 45 species, I only knew about 2 or 3 of them. So this is after 50 years of working with these plants. I can go to a place and find a lot of species that are new. The little area that I have on the map called Chocó, Quibdó, that's up in the upper center, where that blue arrow is, that was a spot probably no bigger than the size of this room. I spent 3 days in this one spot, continuing to find new species all the time. And after 3 days, I thought, my God, I better go on, because there's probably some other places that are equally rich. So after 3 days, I gave up and went on down the road. But it's quite astonishing how species rich a single area can be. And they're not always in dry forest, in wet forest. That picture in the lower lower right-hand corner, that's a dry forest, and we're finding new species there. Of course, not in such great numbers. High habit diversity. Habit means the way it's growing. For example, they may be tuberous plants with a potato-like tuber, or they may be rooted aquatics or floating aquatics like the limnoideae or pistia, which are just, they float across lakes and rivers on the edges of rivers. I call them little mouse rafts. The epiphytes can be of various kinds. For example, Anthurium gracile, it occurs very high up in the forest, but the only way it can get up there, grow that high, is to have a means to get nutrients, because you can't get nutrients on the tips of the upper branches. That's always because nothing falls onto you. But there they're living with the ants, and the ants are providing the nutrients. The detritus from the ant nest is providing the nutrients for the plant. Then there are epiphytes that are secondary epiphytes. A bird deposits the seed on a branch way high up in the tree, and it grows very slowly until it can get roots sent to the ground, and then it becomes a primary epiphyte. So it's starting backwards, it's starting in the tree and working its way to the ground. Ecologically, they're very diverse as well, ranging from tuberous plants that grow in very dry habitats, plants that are completely submerged. This skunk cabbage, for example, is actually a plant that emerges through snow in Michigan and northern Iowa and so forth. It's thermogenic, so it actually produces heat that can melt its way through the snow. And even at that time of the year, it gets some flies. Flies come out just as soon as they can get out of the thaw. And it attracts flies with this very strong, stinky smell. The diversity of size is just as amazing. If you think of limna, which has leaves less than 1/3 of a millimeter across, there's a microscopic picture of it here showing the whole plant. And then you compare that with the leaves of like this alocasia, where they're more than 2 meters long. Some of them are 2 meters wide and almost 3 meters long. Another thing that is quite variable is the size of the actual flowers. I mean, look at Pistia stratioides with a flower less than 1 centimeter long. The whole inflorescence is less than 1 centimeter long. And then you got Amorphophallus titanum, which has got the largest inflorescence of any plant in the world. That plant is one that is of great interest to horticultural purposes because it attracts a lot of people. It has a very foul aroma, but it flowers very infrequently, and when you do get it into flower, a lot of the public comes in and pays money to see it. God knows why. We managed to set fruit on this thing, and the fruits are viable, so they can be dispersed to other botanical gardens. There's a high rate of high diversity in the leaf shapes as well, from very divided leaves. Mind you, this is a single leaf in the upper left-hand corner, a single leaf with lots of divisions in it. Then you have things like this with these discusly arranged leaves in the middle picture, ovate, narrowly ovate picture. Sometimes they're sinuate along the margins, sometimes they're perforate, such as in Monstera, and Eracema has these highly divided leaves with a palmately lobed leaves. Here's a whole array of different leaf shapes, and it's important to note that for every large genus, it has every conceivable form of shape of leaf. So the leaves by themselves are fairly useless in defining the genus. Every genus has a wide array of leaf shapes. So what does separate them? Well, it's mostly the flowers, but I can separate nearly every genus of neotropical plants with just a little strip of vegetation between the midrib and the margin. Just looking at the characters in the leaf itself, I can distinguish all the genera. So there are very good characters. In fact, throughout the family, the neat thing is that you don't need flowers. I tell people, collect the sterile plants. I can still tell you what it is, because there are enough characters in the sterile plants that allow you to identify them. There's a lot of diversity in reproductive systems. Everywhere from these fly-pollinated things on the left to anthurium-pollinated things on the right. Amorphophallus, they always have a strong scent. They're generally pollinated either by flies or by some beetle that likes stinky things. Some species attract fungus gnats, others flies, and others beetles. Always different genera. The flies and the beetles never coexist in these same pollination systems. Many of the species have unisexual flowers, and by that I mean they're monoecious plants. They have the female part at the bottom and the male part at the top, and in the middle you will see a little section here. These are sterile male flowers, and they're nearly always high in lipid content, so that they have a nutrient value. And when the beetles are in these flowers having sex, they have to have something to eat, so they eat these sterile male flowers. They spend 24 hours in there. They come in the late afternoon, about at dusk. They stay until the next day, and then they fly off to another orgy and another flower. And by the way, the flowers provide them with the stickiness, because if they weren't sticky, they wouldn't get the pollen adhering to them, because beetles are very slick, and they get this sticky resin on their carapace, and then when they leave, they get covered with pollen. Anthurium is often, I think, mostly pollinated by bees, and they are attracted to the inflorescence by a strong scent. And very often these are extremely temporal. They will flower just at a particular time, like 7 o'clock in the morning to 7:30, and another one will start at 8:30 to 9. And I had one in my office a few years ago that always had flowered. It started smelling, I could smell it at 11:30. It went from 11:30 to 1, and I wondered why. It's strange timing. It's because it occurred in a cloud forest where it was raining all morning, and it began raining again in the afternoon, but the only time it wasn't raining was 11:30 to 1. So the plant had evolved a pollination system that allowed it to attract bees. The picture on the right is a xanthosoma. These little sterile male flowers at the bottom, again, they're high in lipids, and the beetles eat these. Now, the primary use of aroids, when you say, well, what's the value of this plant family? It's primarily for ornamental purposes. If you take all the potted plants sold in North America every year, half of that list is always aroids. Now, they're kind of boring things because they don't try to grow the really super hard to grow things. They just sell the junky species. But they're the sorts of plants that you can put in your bathroom, forget to water for a month, and it still looks just exactly like it did when you put it in there. McDonald's puts them in their stores, and they're very easy to grow. They're very hard to kill, and that's why they're popular with people. The other secondary use is that they are used for crop plants. If you take the more heavily used ones, this Certosperma mercuzii is not used in the New World. It's only eaten in the Old World, but you can imagine this would provide quite a bit of food. This is a massive tuber, so they could probably eat for a week on this one single tuber. And then in the New World, we have a lot of Old World genera, like not Certosperma, but Alocasia and Colocasia. They're commonly used as plants, and I'll later show you a picture of what I found in Peru. And then in Africa, they're using the New World when they're using xanthosoma as a food crop. And in Hawaii, they have about 60 species, or 60 forms or varieties. We'd call them forms. They can recognize that many different types of Colocasia esculenta. It's all one species to botanists, but they recognize the chemical differences in every one of these species that they use in their food crops. And they even eat the leaves. They call this poi. It's a very traditional Hawaiian dish. The story that I was going to tell you, last summer I went to a little place called Puerto Bermudez. I'd never been in Ukiale. I wanted to go down there and see what was like. When we got there, there was a brand new road leading into the town. But this town had been isolated for probably 100 years. And yet they had all these plants growing. Every house had all these plants growing. So all these aroids were being used as food crops in this little jungle town. It's quite amazing. Now, where did aroids come from? We know that they evolved sometime in the Cretaceous, early Cretaceous, and they formed from the Alispetales, along with a little family called the Tophilaceae. If you look at aroids, these are the aroids in black. No, I'm sorry, up there above, and their sister groups are clustered with them. The family evolved sometime between 128 and 133 million years ago. So it's one of the older plant families, even vying for a position against some of the dicots that we thought were much, much older than aroids. So monocots are not necessarily a lot older than dicots, as we have always thought for many, many years. All 8 subfamilies of Araceae evolved before the KT boundary, which is the Cretaceous Tertiary boundary where there was a lot of extinction. This was the period of time when either meteorites or volcanoes snuffed out the dinosaurs by choking off the sun and leaving food crops and leaving the vegetation die. So the dinosaurs disappeared at that time. Already the aroids had existed before the dinosaurs, in other words. Now, they arose in Laurasia, which is a part of a breakup of Spongia, this superplanet that was present in the Jurassic. In 120, this is really fuzzy here. Anyway, during the Jurassic, 100, I can't read this, so you're going to have to, can any of you read the numbers? Anyway, this is what happened to Pangea. Broke up, Laurasia is here, Gondwana is here. Gondwana began to break up centuries later. You can see India floating off to the right here. It's going to smash into Asia. Africa and South America were still hanging together. Antarctica was drifting to the south. Australia was drifting to the east. It eventually gets carried off completely separate from the Antarctica. India eventually smashes into the Asian continent, and it's still pushing under the Asian continent. So even though the Ariaceae evolved first in Laurasia, they quickly migrated to this other part of these other continents, because they occur in Africa. Africa probably had a lot more at one time, but it got dry and lost a lot of them. But in South America, we have evidence that they really went wild there. Probably because of the rise of the Andes. When the Andes rose, it created millions of habitats, and those millions of habitats resulted in many, many species. That's why the neotropics is so much richer than the old world tropics. And I'm going to talk about that in a moment. The number of species of Ariaceae are presently at a little over 3,600 species, with a conservative estimate of 6,500 species. But I'm going to show you later that I believe that there may be more species in Colombia alone than we have in the entire world right now. You may not believe this, but I think it's a convincing argument. There were 8 subfamilies, and those 8 subfamilies are arranged in a cladogram or a phylogenetic tree here. A phylogenetic tree is like your family tree. It's the degree of relatedness is exhibited by the way the branches come off. The more primitive groups are up here at the top, and then the more advanced groups are down at the bottom. These groups down here have unisexual flowers. They're naked. They have no protective covering over them, like Anthurium. Anthurium have tepals, these little structures that protect the ovary. Down here they're naked tepals. And they also have, laticifers. They don't have sporopollenin, and they have no apertures in the pollen. So there are very big differences between those unisexual flowered plants and the bisexual flowered plants. We've done several phylogenies. Interestingly enough, one of the first phylogenies that was done was by our own Mike Graham, as his PhD thesis at the University of Massachusetts. Mike Graham was studying pollen, and he did a very exhaustive study, not using a computer, but just wrote information. It was a morpho-anatomical cladogram. And he began to question the validity of the so-called Schottian-Englerian system, which was around for 100 years. This was based on H.W. Schott and Engler. It was their system. And so he began to cast doubt on whether this system was accurate. And he gave his thesis was presented at the Second International Aroid Conference in Petersham, Massachusetts, by his professor, Jimmy Walker. And when this was all everybody's slack-jawed, they really couldn't believe that what they had seen. And Joseph Bogner, this German guy who was really the world's authority on Ariaceae genera, he got to thinking, of course, that maybe Graham was right. And so he got together with Dan Nicholson, and together they worked out another system, which was slightly different than Mike Graham's system. So then Jim French did a real molecular study based on restriction site analysis, and Simon Mayo using some of his ideas. Jim French began to show molecularly that Mike Graham was right, and then Simon Mayo did another morpho-anatomical study, which used some of Jim French's restriction site data, and so he came up with basically what we have now, which is the system that we have today. My student my student Claudia Enriquez did something called next-gen sequencing, where it's a very powerful tool, and she came up with all these same subfamilies. So basically, she's just confirming what already was formulated by these other earlier systems. So very slowly, or rather very quickly, we came to a realization that the relationships between these existing groups are as they are today. Monica Carlson, my student, is also a molecular biologist, but she and I and a couple of other people are working with Anthurium, which in my opinion is far more interesting. The subgeneric classification of Anthurium, I think, is a great deal more interesting than the the whole family, because that's pretty well known by now. But we're having great difficulty getting good results with Anthurium at the sectional level. The higher your group, the easier it is to separate the differences. Claudia also did another DNA study with the protein coding genes, but she came up with basically the same story, the same grouping of genera, and the same grouping of tribes and subfamilies. The Araceae are distributed worldwide, but most of the genera are in Asia. At least there's a lot more genera in Asia proportionally than in the New World tropics. We have only 41, they have 63. 27 of them alone occur in Borneo. Africa is relatively poor in genera, and it's probably because it dried up. It's a lot drier continent than either Asia or New World tropics. Most of these things are in tropical areas, and only 6 of them growing in the north temperate regions. If you think about the genera, and you cast out a pie chart of the genera, you'll find that 67% of the genera are just in the aroideae alone. The Lasioideus and in the Monsteroideae, they do have a significant representation. But if you take the species, if you look at just species, look what happens. The Pothoideae, with only 4 genera, mostly just Anthurium, almost is as large as the Philodendroideae or the aroideae. Now, what I'm contending is that this pie chart is going to look very, very different in, say, 50 years from now, if the plants exist that long. Because I suspect that these 2 big genera, Philodendron and Anthurium, are going to grow at dramatic sizes. I don't think Asia is going to grow as fast because, first of all, Asia is more well-known. It seems to have evolved a lot of genera, but not so many species. The largest genus there is only about 127 species, which is peanuts compared to almost any genus in the New World tropics. Now I'm going to go through the subfamilies quickly. The Gymnostichioides has only 1 species. It's endemic to southern temperate Australia. The Oranthioideae has these 3 genera. Has the 3 genera. What many of you perhaps know, if you live on the East Coast, this golden club occurs in ponds and lakes in the early spring, and the Lysochiton, and Lysochiton is Pacific Northwest. If you're from Northern California or Washington State, you may know that. And then Symphocarpus fetidus, that's the skunk cabbage of North Temperate North America. And each of these last 2 have representatives in Asia. Both of them have a representative in Eastern Asia. Then the Lemnoideae, the so-called formerly Lemnaceae, these are floating little scum on the ponds, and they're practically invisible to the naked eye. You have to have a microscope almost to see anything on these plants. But nevertheless, the molecular data puts them flat in the Araceae, right smack in the middle of the Araceae, but not anywhere close to Pistia, the other floating aquatic plant. So all of these groups here are probably evolved from aquatic ancestors. In fact, the Araceae was a strictly aquatic family in Asia. That's why it evolved with the Elismataceae, which is another aquatic family. You might imagine this is because in the early days, most of the earth was covered with water. It was a very swampy place. So any plant like growing a tuber or something, it would never survive in such a habitat. So all of these things a lot of them have reverted to their aquatic state. The Pothoideae is a huge group, but only because of Anthurium. The genus Pothos is relatively small with only 57 species, and these other 2 little genera, they have only 1 species each. Anthurium has between 2,000 and 2,500 species conservatively, and I think it's probably got at least 3,000 species. The Monsteroideae is a group that has a kind of a big split in it. You've got plants with intact entire leaves like Spathiphyllum, they're terrestrial plants, and you've got a few epiphytic plants like these, or climbers, with long banana-like leaves, and stenous formation, which have practically no venation. These have long leaves with no perforations or pinnately lobed leaves. And then the rest of the plants are all epiphytic climbers, hemiepiphytic climbers with divided leaves or perforated leaves. The Lasioideae, they're largely swamp plants. The vast majority of these are swamp plants, except for the genus Dracontium, which are nearly always in seasonally dry habitats. They have a big tuber that allows them to survive during the dry season. The Caloideae is a group of plants. It's only one species, but it's very, very widespread. It occurs all across the northern temperate regions. If you look at this next picture, you'll see that Calla is distributed outside of the boundaries of the Poethoideae. I mean, outside the boundaries of the Aroideae. So it was ostensibly a part of the Aroideae, but it doesn't fit well because it has bisexual flowers, it has pollen with apertures, it has this sporopollenin, but on the other hand, it has leticifers, which are unknown otherwise in the other groups, the more primitive groups. So it's an oddball, and that's why it's in its own subfamily. Then you have another small subfamily, the Zemiococadiae. This thing is from South Africa, and there's only 2 genera, and with very few species. These are also unusual because they are in a group that have unisexual flowers, but they have tepals. They have these little coverings over top of the anther, over top of the flowers. So that's another oddball thing, which doesn't fit anywhere well, and for that reason, it fell into its own subfamily. So we got 2 subfamilies, which were basically a couple of species. And then you've got this. This is Aroideae. All of these, this thing has, I think it's 27 tribes. These are just the tribes. So within these, there are also a bunch of species, a bunch of genera. So this group here have all these different tribes, may have one species or they may have many. Here's a Stylochaetanae, which is from Africa. And it's kind of interesting because it very often has the inflorescence submerged in mud or in dirt. And the pollinators must crawl around in the soil to get to these plants because sometimes the flower isn't even available to them. We don't know what pollinates it, but it must be some kind of a creeper in the ground. And then the Dieffenbachii, this consists of 2 groups, Bognera, which is a new genus that has only 1 species, and the genus Dieffenbachii with about 120 species, all in the neotropics, ranging from Mexico to Argentina. The Spathocarpii is a group that has strange, the anthers are often in stalks, like you see on the 2 pictures on the right, and these stalked inflorescences with pollen on the ends of the filaments. But Croatiella, this is, by the way, do I have any Croats in here? Anyway, this is named after a Croat, so you can brag about that. This one does not have the same characteristics as Teckarum and Gorgonidium. And then there's the Philodendroideae, another enormous group. Philodendron has at least 1,000 species, and it's divided up into 3 subgenera: Pteramecium, Micronostigma, and Philodendron. And Philodendron, the subgenus Philodendron, makes up about 95% of the total. But those other 2 groups are also quite large, but they're dwarfed by the size of the Philodendroideae. And there's also one other little genus in here, which I recently revised, this Adelonema with 13 species. This was formerly known as Homalamina, but the molecular studies proved it to be distinct at the generic level, so it adopted one of the older synonym names was Adelonema. The group from which it was split off was the Homalamina in Southeast Asia. Homalamina is a group that has 98 species, but it's also the genus in Asia with the greatest potential for growth. There are potentially a lot of new species going to be coming out of Homalamina in Southeast Asia. We have also Anubias, the Nubidae, which is based on one genus from Central Africa, tropical Africa. These are largely swamp plants. There's only about 6 species in Anubias. Schismatoglottidae, this is another large group in Asia with 7 genera. And this group had a genus in the New World, or had species in the New World which were found to be molecularly distinct, and so they became the genus Philonocian. So you got 2 pictures of Philonocian on the right-hand side, and this is Schismatoglottis. They have these typically margined, beautiful leaves. And then there's another group of Schismatoglottidae, which are largely rapiculous, or they're plants that grow along the edges of streams and in waterfalls. They're always associated with standing water or with running water. And you'll note that the spathe falls off very quickly. Notice in the upper 2 pictures, the spathe is on, intact, and then the blade of the spathe falls off, forming a little cup. Those little cups are important because they occur in situations where water either drops in or pours in from waterfalls. And as the seeds mature, the water pouring into this cup blows the seeds out into the water. So it helps to disperse the seeds over a period of time. Every plant species likes to disperse its seeds over a period of time rather than all at once, because they may all fall into the same hole and not be dispersed very well. Another group of strictly aquatic plants is this tribe Cryptocoryneae. It has only 2 genera, both of which are more or less submerged aquatics. They occur in tidal ponds where sometimes the water is out and sometimes the water is in. Maybe they flower on neap tide, or they flower when the water is the lowest. Because their inflorescences are kind of weak, the plant sort of waits until the water leaves and then they flower. But they're pollinated by flies, and they have these little traps in between the spathe tube and the spathe blade to capture the fly after it gets in there, so as to hold it in there a little bit longer. Flies are rather stupid pollinators, but they fall into traps and then try to get out. They just bounce around. You've seen this in your own situation, right? Where they're always trying to get out, banging against the window instead of going to the door. They just bang, 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 bang. Well, this helps them to spread the pollen. So the more they waltz around in there, the more pollen they get on the stigma. And then eventually they hit the hole by accident and leave. Then we have the Zomicarpi, another small group from South America. This is so rare that I already saw the thing once or twice. It only has it's 2 genera and I think a total of 5 species. It has little hooded inflorescences like you find with Dracontium sometimes, and with Jack in the Pulpit, for example. These little protective hoods are probably for protecting the pollen, because when you have a little hood over your spadix, the pollen doesn't get wet when it rains, and therefore the pollen stays dry and the insect can carry it to another plant without losing it. If it's all washed off in the first drizzle, then it's of no value to anybody. The Caladiae is a group that many of you know as the genus Caladium. Sometimes you grow these in your yard. They're very colorful. This particular plant here is one that was described from a sterile plant, and for more than a century it was known only as a sterile plant. It had never flowered, but someone hit it with some colchicine or something and forced it to flower, and so now we know what its inflorescence looks like. This Caladium palacioanum was described by myself, but from a population in a little narrow canyon in southeastern Ecuador. And Lindenii is a common houseplant. Well, it's common to me at least. The Nephtidae is a group that occurs in both Africa and Asia, but most of the species are in Africa. They occur as an understory herb. They have relatively few male flowers, few female flowers, and they have a lot of male flowers. And then the berries, I don't have a picture of the berries on here. I did have, but we lost that picture somehow. Well, here's the same sort of thing. It'll look very much like Aglaonema with just a few fruits at the base. It loses its spathe altogether, and the berries are present there, colorful red berries. This particular group, this Aglaonema, this is a relatively small genus with 25 species, but very, very beautiful species. Nearly every one of them has some kind of color affection on the leaves, and they're very highly sought after by horticultural groups. The Colcaceae is a genus from Central Africa, and it's probably the group that has the greatest potential for new species of aroids in all of Africa, because I've seen herbarium specimens, many herbarium specimens that have not been identified yet, and it probably means that they're an undescribed species. The Montricardiae, this consists of only 2 species, one basically North American, one is South American, with some overlap in Venezuela. These have large fruits that are buoyant in water, and they are capable of floating away, and then once they get waterlogged, they sink to the bottom, and that's how they disperse their seeds, further down the river or in another part of the lake. The group of Xanthidaceae consists of just one genus with 8 species, and these are heavily used horticulturally because they've done a lot of breeding with these, producing some very interesting color forms. I've tried growing these in my own yard, but they don't seem to last for very many years. There's a little bit cold tolerant, in other words. And this little thing from Central Africa is only known from one species, this Calopsis volkensii. It's a beautiful little plant with cordate leaves and big white broad spathes. And the Thomsonii, they should have called it the Morphophalae, but in fact, Thomsonia was used first, so they had to use that name. Amorphophallus is a group that's quite large. It's one of the larger genera in Asia, and it's very, very diverse. I had only space here to include a few pictures, but you could do a whole lecture on Amorphophallus by itself. It's an incredibly diverse group of plants from the standpoint of its flowers, and especially its spadices. This Aerophyte is a small group from Madagascar, endemic to Madagascar. Joseph Bogner was the person who went searching for these things, trekking into all parts of Madagascar to find these rare endemic species. So we know quite a lot about them today, and quite a number of them in cultivation. This is a strange group too, the Peltandriae. Peltandra has a Peltandra, which is Peltandra virginica here in North America. Its closest relative is in Madagascar. So how do you explain that? And they're very close from the standpoint of molecular studies. So we know they're very closely related, but how they got separated is amazing. And then the Ericeae, this is another group that perhaps many of you know. This is Jack in the pulpit. We have Ericema triphyllum, and we also have Ericema. Oh no, I'm sorry. Sorry, not Ericema. This is a Mediterranean species, Mediterranean group. There are only 3 species in the group. They're all depicted here. Ericeum are pollinated by flies. They have, foul-smelling flowers. They flower in, just like this next one. They flower in the winter when it's wet, and in the summer they completely go dormant with just a tuber to sustain them. And you'll see this unusual thing. This is a trap. These flies are attracted to this foul scent. They get in here, they get trapped in here, and they affect the pollination before they can escape. This is Ambrosini, only known from this one species, Ambrosina bassii. Another larger group in the same vein, the Aerae. There is quite a number of genera in this group. They're mostly Mediterranean, but they get as far east as India and even Vietnam. This plant, which was named Croatii, was described from material in Vietnam. So we know that they get as far as Vietnam. Some of these I've grown this Soromatum venosum in my yard. It's attracted, flies are attracted to it in the early spring when it's flowering, and it even sets fruits in my yard. You can see that not all aroids are growing in wet places. Look at this habitat. It looks like a desert. This is in Iran. So this group is fly pollinated, purplish inflorescences, stinky, and tuberous. And then this group, which is one I was trying to talk about, is the Jack in the pulpit, our Ericema triphyllum, Ericema draconopterum. This is the other Ericema in North America. And then we have one species in Mexico, and that's it for North America. There are a lot of species in Asia. Most of the species of this group are in Asia. And then Pinellia is another group that's closely related in the same tribe. Pinellia is another group of related terrestrial plants. I grow one of these in my yard. It's actually quite a weed. It's attractive, but it grows. It gets into everything. It's a very good dispersal somehow. Maybe the ants carry it away because you find it coming up in cracks in sidewalks and anywhere. Now the Colocasioideae is another group that's huge in terms of numbers of genera and numbers of species. These are also heavily cultivated. Colocasia esculenta is one of the plants that is growing as an edible plant. Many of them are very attractive. I don't have all of the more attractive ones here, but a lot of them have colored patterns. I grow this mycorrhiza, and I grow Colocasia esculenta in my yard. It doesn't get this big because I have to dig it up every year. So if I had it in a tropical place, that it would grow taller than my house. But as it is, it only gets about 2 meters tall, then I have to put it away. So it never gets terribly large. And finally, the Pistii. This is the group that is floating aquatic. It has tiny little flowers and unrelated to the Lemnoideae, but with the same characteristics as all the other aroideae. Now I'm going to switch to another topic altogether, and I'm going to talk about what we have accomplished here at the Missouri Botanical Gardens. This is a list of the genera that I have worked with, and the increases in the size in terms of numbers of species in those same genera over the past 40 years. I picked 40 years just out of my hat, but that happens to coincide more or less with the same period of time that I was working on aroids. And you can see that there's a lot of increase in the size of these genera, meaning that, in fact, there's a lot of potential for growth because most of these species that I have discovered are actually from Central America, not in South America. And South America is much larger than Central America, as I'll point out. So I want to make a case for a much larger Araceae. We've developed these lucid keys. It's based on lucid technology. We've put all the character states of every anthurium into this key, and every philodendron, and every dracontium. We've made about a half a dozen of these keys now. And this enables you to more predictably identify a plant because the computer is telling you all the possible choices. It's telling you all the possible species that it could be. And the computer is dropping things out. I'll show in a moment. So we have about 1,530 species in this key. In Philodendron, we have about 600 species. And what's the astonishing thing is we have boxes and boxes and boxes of plants that won't key out. I haven't had time to deal with them to see whether they're really new species or not, but they do not key out in the lucid key, so they don't seem to be species that are in the key, which are the species that are described. So that indicates to me that they're likely to be new. It just takes more time to sort them out. The lucid key operates, you have a bunch of characters over here. These character states or features, you can choose the character states by just clicking on them. And as you click on them, the species on the other side of the thing drop away. So you start off with 1,530 species, and every time you select a feature, if the plant in that group doesn't belong there, it falls out and it goes down here. So this is just an example of how we keyed something out. And here are 6 species left. I think there's 6 or 5, I don't know. Anyway, there's a few left here. So those are things that you can get your mind around. 6 species, these are the most likely species compared to the thing you're looking at. And at that point, we can actually go to another tool. We've made a determination tool. Steve Aylward, one of my volunteers, actually made this for me. He put all these species into files so that we can, in a moment, pick out any name and get the description, get the full description, get the type photograph, pictures of living plants, and you can see quickly whether this plant looks like the plant you're keying out. So it's a remarkable way of making identifications because we have all these tools accessible to us now, largely thanks to volunteers. Another way to approach this subject is to look at existing floras and compare them with other places that are unknown. For Central America, we have a checklist. This is just the tail end of it. But the checklist of Central America, we have, I can't even read it, but it's too fuzzy. Anyway, we have nearly 800 species. Colombia, in their checklist, has 820 species. Panama has 661 species or something like that. So here's Panama with 661 species, and we know they're good species because we've worked in these things for years and years and years. Panama perhaps has 10% more species, so the 661 may be even higher. But to compare it with Colombia, it makes Colombia look too weak. Colombia, in fact, has too few species. And unquestionably, this is the case. If you look at these numbers along here, you'll notice that the numbers of species increase as you approach South America. Every country you get closer to South America, they get larger and larger and larger. Well, what happens next? Colombia has to have staggering numbers because it's not only 15 times larger than Panama, but it's probably 10 times more diverse in every respect. So this, to me, is conclusive proof that Colombia has vastly large numbers of species than we are presently dictating. For example, if we take the 622 species in Panama, 80% of the flora of Central America, and we compare that with the 820 species from Colombia, this is absurd. We have to have many, many more species in Colombia. So it's a matter of getting out there and finding these things. The endemism also increases. As the species diversity increases, endemism also increases. So Colombia probably has a lot of endemics too, meaning it's not likely to have the species that are in Panama, some of them, of course, but it's likely to have its own endemic species. So I think that Colombia may have 8,000 to 12,000 more species than currently are known. That's more species than exist currently in the entire world. And the collecting program has essentially stopped everywhere. There's very little collecting going on anywhere. And yet this is at the same time that this extinction is going to take place, because everything is going and we're not doing anything about it. We're not collecting it. The experts should be encouraged to get out into the field to collect, because they're the ones who are most likely to find the new species. You can send somebody out for a few days and they come back. They might come back with the commonest thing possible. There was a study in England a few years ago that showed that of all the species that have been described, they were described by only about 3% of the species, 3% of the people who describe plants. And they referred to these people as mega collectors. And they had three things in common. They collected throughout their entire career. In my case, it was like 58 years. They collected many different places. I've collected 142 countries, or I collected 142 countries. And they also had a reason to go to the field, a compelling reason to force them to go back again and again and again, to endure the hardships of collecting, because they had something that they wanted to find. They knew there was something there for them to see. This collecting is no longer considered important. And we have too few students studying floristics. We have too few people collecting. Whereas the floristic studies is where everything begins. If you have people going out there for a reason to collect, it's difficult to say, just go out and collect. If you give them, say, go out there and collect everything in this area, and let's do a summary of what we got, then there's a compelling reason to do it. And if you publish those studies, then they're going to feel important, and they'll do more of them. We aren't doing this at all now, so it's very pathetic that we're killing the goose that gives us all of this information. So there's also problems with getting permits. It's very difficult. A lot of people just give up, don't try. Many people don't collect anymore because they're too old. But if they had a compelling reason to go there, maybe they'd go. You know, I went last year, just like I've gone every year, and I'm 80 years old. So, I mean, you can do it. Now, this is what happens when we just sit aside and do nothing. This is a common situation. Here's a forest. You can see this has been cut off. Here's some virgin forest. Here's some pasture. You can see this everywhere. You can see, but that next year that's going to go too. And then you see the forest being burned here. Fires going everywhere. And then you got this, nothing at all. Hyperhena rufa, nothing else growing. No other plant will even survive on this damn forest. This soil lacks any kind of nutrients. A few ferns and a few very strange grasses will grow in these places. So this is what's going to happen. We need to spend more time dealing with this. The destruction is taking place despite any efforts at conservation. Most of these reserves aren't in. Most of the plants that exist aren't in parks and reserves, so we couldn't reserve them anyway. They're private lands. They're held and owned privately. During my career, nearly everything has disappeared. All the places where I ever went, including where I went last year, it's gone. And everything goes just as fast as you can see it. They build a road, next year it's gone. And with the endemism that's prevalent in the Area Sea, that means that every spot has endemics, and therefore it means extinction. The Missouri Botanical Gardens has been traditionally a place of discovery. We're very important in this process of discovery. Every year, when you look at the group of species published new to science every year, the Missouri Botanical Gardens has a high place in this list of order of people who have done it. So, you know, we have a tradition of describing new species. And we have a tradition of fieldwork and the importance of fieldwork. Peter Raven used to say, "How long have you been here? Why aren't you in the field?" You know, he would encourage you to get out of here, you know, go collect. The importance of exploring new areas is another thing. I have a little study that we made on Cerro Calan. It's a place in northern Peru. It was just a simple little place that people could walk to. Terrible path. I tried to get there and it was impossible. Mud clear up to your knees. I finally gave up. But people did go in there. LSU sent a team of about 6 or 7 guys in there. They spent 4 months there. They collected all kinds of birds, mammals, reptiles, plants. That place, after 30 years, nothing. There's nothing left at all. So, the Area Sea has vast potential for producing new species. We need to increase our collecting activities. And these floristic studies should be encouraged because that's really where you end up getting plant collections. And tropical floras are, the expertise on tropical floras is generated by these people that can get to them. And this is perfect for Latin American students because most of them don't even have money for bus fare. But it's cheaper to do a little flora in their local area than any other kind of studies they could do. And these are very rewarding studies for Latin American students. I know a lot of Latin American students. I always encourage them. Look, you got this hill over there that's still forested. Why don't you go collect there and collect all the aroids there and do a little study. We publish these in Aroidiana because we think it's very important to do. And more of this kind of thing should be encouraged because we're cutting off our valuable source of plants. I want to just finish with some closing remarks. You can see that we already know a lot about Araceae, and I believe that I've advanced our scientific knowledge of this family in a major way. God has blessed me with abundant energy, strength, and good health. I didn't lose a single day of sick leave in 50 years. I have the support of a very capable and loving wife who allowed me to traipse around the jungles of the world for months. Since I didn't take vacations, my kids went on to vacations with their mother or their Aunt Rosemary. The Missouri Botanical Gardens is a marvelous place for work. The horticultural staff supports the study of thousands of living plants. The research division provides tremendous help in processing, cataloging, and studying collections. I've also had the good support from the National Science Foundation and the National Geographic Society over the years. I've got collaborators here and throughout the world who work together on this family. The International Aroid Society is a very active and enthusiastic group of mostly horticultural people and I've served on their board for the last 40 years. Finally, I have a dedicated assistant and many dedicated, talented volunteers, now numbering about 40. These have aided me in identification and describing new species, preparing manuscripts, constructing new lucid keys, new websites, digitizing and processing digital images. In the past 2 years, I have traveled throughout South and Central America and Europe on an OPUS grant, determining plants, taking pictures of undetermined collections, and engaging with young botanists to inform them about the wonders of aroids and to assist them in their own projects, and finding others to join the research that needs to be done. You can see that we have much more to do, and adelante, we'll continue this. Bye-bye. Thank you very much.